In this video, we're going to be talking about membranes and transport. This is how the cell gets molecules and different ions into and out of the cytoplasm and organelles. This is how the cell moves things around. So in general, there are two types of transport. There's active, and then there is passive. Now, the difference is whether you use ATP. ATP stands for adenosine triphosphate, an organic compound that is composed of an adenine group with 3 phosphates attached. And the way that this works generally is that you have the A and then you have the 3 P's. And when you break the bond in between these, the second and the third P, you are releasing a lot of energy. The P floats off and this becomes adenosine diphosphate, the di meaning 2 of them, plus P, which is that extra phosphate group. And you get a whole bunch of energy coming out of there. And active transport basically uses energy, but passive transport does not. So active uses energy, and passive does not. Passive transport does not use energy. So when we talk about active transport, there are a couple of different types. There are protein pumps, endocytosis, and exocytosis. So a protein pump is there. You have the lipid membrane. You have the phospholipid membrane, the bilayer, with the polar heads facing out and the nonpolar heads facing in. And what a protein pump looks like is it's just this giant intermembrane protein that looks like this. And what happens is that molecules, they enter the cell here, and they are forced through this side. Now, before we get into this concept, we have to talk about something called diffusion, about hypertonic, hypotonic, and isotonic. These are 3 very confusing terms for biology students because they are very conflicting, and it really depends on your reference, your frame of reference. Which one is this? So, if I have a cell inside of a solution, if I have a cell inside of a beaker and I fill the beaker with some water, I have 2 molecules in there, 2 solutes in there, and I have 5 outside of the cell. Well, the term hypertonic means more stuff. Hypotonic means less stuff. Isotonic means same stuff. Put quotations around those. What it really means is more solutes, but we're going to refer to it as stuff because it makes a lot more intuitive sense. Now, which one of these has more stuff? Go ahead and pause the video, think about it. Okay, so which one of these has more stuff? Well, it would be the solution. Now, if we look on our cheat sheet, we see that more stuff means that it's hypertonic. So, hypertonic. Now, you may have gotten confused. We're talking about the solution with respect to the cell. So we're deciding which one between the cell or the solution has more stuff. Okay? So if we refer to this whole thing as one system, we say that this is the system. Cell plus solution. Okay, that is the system. Okay? If we had two cells in if we, let's say we had a system where we had like two cells, we had a cell there and a cell here, then our system would be two cells plus a solution. Okay, so that's our system. Now, what we're finding is which of these is hypotonic in the system, okay? So within this system, the cell plus the solution, which one of these is hypertonic? Well, we're looking for the one with more stuff, and we see that it's the beaker. And which one is, that means that the other one must be hypotonic, because with respect to the system, with respect to everything else that's going on inside that beaker, it's this cell. So what we have here is a hypotonic cell. We have here a hypotonic cell in a hypertonic solution. Okay. Now, going back to active transport, I'm going to talk about when we use it. Now, we use active transport when going from a hypotonic to hypertonic. So when we want to go, when a solute wants to go from a hypotonic solution where there's less stuff to where there's more stuff, so less stuff when you want to go from where there's less stuff to where there's more stuff, you need to use active transport. Why? Because it actually requires energy to go from less to more. So let's say that this solute here decides he doesn't want to be in the cell anymore. If he wants to leave and come out here, he's going to go through active transport, using up energy in the process. So the first way that that a solute can achieve that is by going through a protein pump like this one, which takes the molecules, takes some of this adenosine triphosphate here, and actually uses it to force molecules from where they're less concentrated to where they're more concentrated. This process here can happen in reverse too. If there's more over here, then we can say we can take some of. If there's less over here, then we can take them and force them up here. So this process can happen either way. It's always going from where it's less concentrated to where there's more concentration. Endocytosis and exocytosis are basically doing the same thing. Now, what happens here is 4 stages. Actually, a number of stages, I'll say that. Let's talk about endocytosis. Exocytosis basically is the opposite of endo. So, first thing that you have is a mass of solutes here, like a whole bunch of them. So the first step is it looks like this. The second stage, in the second stage, the cell the molecules come very close to the cell. In the third stage, the cell membrane transforms into a very nice little bubble for these solutes. They come close to the cell membrane, and it just sort of like looks like a sideways Pac-Man. If you imagine Pac-Man. Pac-Man is technically going through the process of endocytosis when he's eating all these whatever solute particles. What he does next in the last stage is the cell membrane. Sorry, I made it green. Let's change it to yellow. In the last stage, the cell membrane closes in, and you get this little vesicle there. So in part 2, we're going to be looking, and exocytosis just goes the opposite way. And in part 2 of this series, we're going to be looking at passive transport.